Welcome back to the deep dive. I'm your host. Today, we're doing more than just discussing a topic. We're basically cracking open a potential blueprint for how we handle future pandemics. Think back to, the toughest days of COVID-19. Now, imagine a device small enough to fit in your hand that could have, instantly cut through the confusion, turned fear into, well, focused action. That's sort of the promise we're digging into today. Okay, let's unpack this. We've all felt that urgent need, right? For rapid, affordable, accurate ways to detect pandemic viruses. It was desperate sometimes. Our source today is this really fascinating scientific paper, and it doesn't just tweak things, it presents a genuinely innovative, almost revolutionary way to spot SARS-CoV-2 RNA. It's got a total rethink of diagnostics. Our mission really is to walk you through this groundbreaking research, show you how it cleverly sidesteps some big hurdles in traditional testing. It could be a potential game changer for global health. At its core, the paper unveils this handheld, low-cost device. Detects SARS-CoV-2 RNA super fast, and it does this using a novel method for making gold nanoparticles. But there's a twist. A surprisingly common metal is key to making it all work.
Yeah, and what's really fascinating here, I think, beyond just the device itself, is how the research tackles those stubborn practical challenges head on. They didn't just engineer, a better mousetrap. It feels more like they designed a whole diagnostic ecosystem. Think about the struggles we all saw globally. The massive cost of testing, right? The agonizing wait times for results, and the logistical nightmare of getting sophisticated gear, maintaining cold chains, especially in remote or under-resourced places. This research, using some incredibly clever chemistry and smart engineering, it addresses all of that at once. We're going to hit some real aha moments today, like the surprising role of just an everyday metal in speeding up a complex chemical reaction, and how something as simple as a color change can be a really powerful, reliable diagnostic signal. It's not just a small step up from current tests. It feels like a fundamental reimagining of making diagnostics accessible. A path to truly democratizing that knowledge about who's infected where, anywhere in the world.
Let's just cast our minds back for a second. December 2019, Wuhan, China. The world first meets what we now know as SARS-CoV-2. Suddenly, symptoms like fever, cough, shortness of breath, severe pneumonia, they became frighteningly familiar. The sheer speed and scale of it all just threw this critical need into sharp relief. Rapid, large-scale diagnosis. Identifying infected people wasn't just about getting them treatment, though that was vital. It was absolutely essential for guiding public health strategy, for understanding community spread, for stopping health systems from just being completely swamped. And this was especially true in really densely populated countries like India, for example. Widespread testing there could quickly become this huge logistical and financial mountain to climb. We saw how testing capacity became the bottleneck, didn't we?
Precisely. Those early days, they really laid bare a brutal reality. Without fast, widespread testing, public health officials were, well, essentially flying blind. Contact tracing became this massive, almost impossible task. Isolation efforts got delayed. The virus always seemed one step ahead. Our existing systems, while scientifically sound, just couldn't cope with the scale of a global pandemic.
Right. And for diagnosing COVID-19, the scientific community pretty quickly settled on the gold standard, real-time reverse transcriptase polymerase chain reaction, RT-PCR. It's praised for being incredibly accurate, right?
Yeah.
It can detect, even quantify, tiny bits of viral RNA in samples like saliva or swabs. It's like finding that needle in a haystack with absolute certainty. But,
Yeah.
and this is where it gets really interesting, and where the limits of this gold standard became painfully obvious during the crisis. The drawbacks of RT-PCR, even with its accuracy, are pretty significant. Expert, can you maybe walk us through those challenges? What made it so hard to deploy at the scale we needed?
Absolutely. Yeah, well, RT-PCR is undeniably precise. Its very nature makes it difficult for that rapid, widespread use we desperately needed. I mean, think about it. It's time-consuming. It requires multiple, quite intricate steps. You've got sample collection, then extraction, then amplification, finally detection. Each step needs specialized lab gear, thermal cyclers, centrifuges, precise pipettes, not to mention a reliable power source, a climate-controlled room. These aren't trivial things. And then there's the cost. The machines themselves are expensive, tens, sometimes hundreds of thousands of dollars. And the diagnostic kits, full of enzymes and reagents, they're costly consumables too. So it created this huge dilemma. If your best, most accurate tool is slow, expensive, and needs a dedicated lab setup, how on earth do you test millions, maybe billions of people in a fast-moving global health crisis? It really highlights that critical gap between what science can do and what's practically accessible, especially for remote places or resource-limited settings where labs and trained staff are, frankly, a luxury. So this raises that important question. If the gold standard is so good but so cumbersome, What are the real innovative alternatives, and how do we design them not just for scientific accuracy, but for global reach and, importantly, rapid impact?
Yeah, that's where innovation truly becomes, well, a lifeline. You mentioned alternatives, and this is where app tamers come into the picture. They sound like a really promising way to maybe revolutionize detecting biomolecules, potentially replacing traditional antibodies. But for listeners who aren't familiar, what exactly are app tamers, and what makes them such a compelling choice for a device like this?
Aptamers are, they're kind of like a molecular engineer's dream, really. They're short, synthetic strands of DNA or RNA oligonucleotides. Basically, think of them as custom-designed molecular hooks or maybe molecular keys. They're precisely engineered to bind with incredible specificity to one particular target molecule. Could be a protein, a small molecule, or, like here, a specific viral RNA sequence. They're picked out from these huge libraries of random sequences using a process called Select Systematic Evolution of Ligands by Exponential Enrichment, which, simply put, is like a very sophisticated molecular treasure hunt to find the absolute perfect binder for your target. Now, antibodies, they've been the workhorses in diagnostics for ages, right? But aptamers offer some really game-changing advantages, especially for a kind of rapid, widespread deployment we needed and still need. First, stability. Aptamers are generally more stable thermally and chemically, and they have a much longer shelf life at room temperature compared to antibodies. This is crucial, often overlooked, but vital for remote deployment or places where a strict cold chain, keeping things refrigerated during transport and storage, is just unreliable or impossible. Imagine trying to distribute millions of refrigerated test kits across, say, a vast hot continent during an outbreak. App cameras basically remove that hurdle. Second is the rapid and scalable synthesis. Unlike antibody production, which can be complex, time-consuming, sometimes needs animals, aptamers can be synthesized chemically, quickly, and on a massive scale, often in just 2 or 3 hours. This means incredibly fast manufacturing and deployment, critical when you're dealing with a new, fast-spreading pathogen. So basically, aptamers give you specificity, stability, and speed, all wrapped up in one neat molecular package.
Okay, so we've got the problem, the big limits of traditional methods like RT-PCR, and now we have a glimpse of this powerful potential solution, these remarkable aptamers. Now let's bring in the other key component of this new detection method from the paper. It relies on the clever use of gold nanoparticles, AUNPs, combined with these DNA aptamers we just discussed. Expert, can you kind of paint a picture for us? What are these tiny gold particles, and how do they work with the aptamers to give us a signal we can actually detect?
Sure, so imagine gold, but not like a coin or jewelry. Think incredibly tiny spheres, scaled down to just 1 to 100 nanometers across, really, really small. At this nanoscale, gold behaves differently. It has these unique optical properties, makes it a fantastic signaling agent. These AUNPs are then functionalized, which basically means they're coded or linked up with those specific DNA aptamers we talked about. Think of it like sticking millions of those specific molecular keys onto the surface of these tiny gold balls. So what does this combination mean for detection? Well, these aptamer-coded AUNPs are designed very precisely to detect SARS-CoV-2 genes through something called complementary hybridization. When the target SARS-CoV-2 RNA is in a sample, it acts like the lock that perfectly matches the aptamer key. The viral RNA then binds or hybridizes to the complementary aptamer sequences stuck on the AUNPs. And here's where the magic happens, where you get the diagnostic signal. When this RNA-DNA hybridization occurs, the solution shows this dramatic, visible color change. It's not random.
It's because the surface properties and how close the nanoparticles are to each other changes, which fundamentally alters how they interact with light.
That's really clever, turning a tiny molecular event into something we can actually see. But you mentioned a key innovation in how these AUNPs are made. Usually making gold nanoparticles is a bit tricky, right? Needs heat, careful control, takes time, adds complexity and cost. But this paper, it has a real game changer here. Expert, this is where that common metal makes its surprising appearance, isn't it?
It absolutely is. And this is where the sheer genius of their approach really comes through. For decades, making metal nanoparticles, gold included, usually involved using liquid chemical reducing agents. These get used up in the reaction, need careful handling, disposal. It's often energy intensive, can take quite a while. But this paper reveals something completely new, genuinely a first disclosure. They're using tin. Yes, like the metal in tin cans. As a reusable solid-state reducing agent for making these metal nanoparticles, this isn't just a small tweak. It's a fundamental shift in how you might do this kind of chemistry. Imagine taking a costly, energy-hungry process and making it almost as simple as stirring something in a pot with an ingredient you can use again. This solid-state reduction is incredibly fast. We're talking nanoparticles forming within just 30 seconds. That speed,
Plus the fact it's easy to scale up makes it really viable for commercial production. It could dramatically lower manufacturing costs. And crucially, unlike those liquid-reducing agents that get consumed and create waste, this solid tin metal can be taken out and reused multiple times. That makes the process not just cheaper, but also significantly more environmentally friendly, more sustainable. It transforms not just the speed, but the whole economics of making these diagnostics. It's a breakthrough with potential uses far beyond just this test, actually.
That is brilliant. Reusable tin, making nanoparticles in seconds, that's radically different. To really appreciate the cleverness, though, we need to understand that color change you mentioned. Expert, can you demystify what's happening at the nanoscale when those gold nanoparticles go from wine red to bluish gray? What's the science behind that?
Right, that visual magic, the color shift, it comes down to this fascinating quantum effect called surface plasmon resonance, or SPR. Don't worry about the complex physics too much. Think of it more like the electrons on the nanoparticle surface sort of humming together collectively. When light hits these tiny gold spheres, the electrons in the metal start oscillating together. These collective oscillations, the surface plasmons absorb and scatter light very strongly at specific wavelengths. And what's really cool is that the color we actually see is incredibly sensitive to the size, shape, and especially how close together these nanoparticles are, their aggregation state. So for instance, when the gold nanoparticles are small and nicely spread out, usually around 17 to 15 nanometers, their SPR peak is around 520, 530 nanometers. That makes the solution look like a vibrant wine red. It's a clear, beautiful color. But if these nanoparticles start clumping together, aggregating into larger structures, maybe 20, 25 nanometers, their SPR peak shifts to a longer wavelength, towards 630 nanometers. This shift totally changes how they absorb and scatter light. And visually, the solution turns into this distinct bluish-gray color. So that change, wine red to bluish-gray, isn't just pretty. It's our direct, reliable visual signal that the target RNA is present.
Gotcha. So the colors basically are diagnostic traffic light. Red means stop, negative, blue means go, positive, roughly speaking. And you mentioned the aptamer is specifically chosen to target the nucleocapsid, the N gene of SARS-CoV-2. Why the N gene? We hear so much about the spike protein, the S gene, especially with variants. What makes the N gene a better target here?
That's an excellent, really crucial question. The spike protein and its S gene, they get a lot of attention because they're involved in how the virus gets into cells, how our immune system responds, and they're often the target for vaccines. But because of that, they also tend to mutate quite a bit. For diagnostics, especially ones you want to work reliably over time as the virus changes, you often prefer a target that's more conserved, less likely to mutate. That reduces the risk of false negatives when new variants pop up. The nucleocapsid, or N gene, is generally much more conserved across different SARS-CoV-2 variants. Plus, the N gene is usually expressed at really high levels during an infection. That means there's generally more N gene RNA around, making it a more robust and sensitive target to detect. It helps avoid sensitivity issues that sometimes affect tests targeting the S gene. It's a strategic choice to make the test more reliable long term. Okay, so let's pull all these pieces together. Let's walk through the detection mechanism step by step. First, we start with our gold nanoparticles, made super efficiently using that cool tin method. These AUNPs are then functionalized, coated with that thiol-modified aptamer. The thiol group forms a really strong, stable bond to the gold surface. That basically anchors our molecular keys securely onto our gold signaling spheres. Second, you introduce the sample, potentially containing SARS-CoV-2 RNA. If the virus RNA is there, it acts as the perfect lock. It finds the aptamer-coated AUNPs, and the viral RNA hybridizes specifically to those complementary aptamer sequences. It sticks right on through Watson-Crick base pairing. This binding effectively coats the surface of the AUNPs with the viral RNA. Third, and this is a key step, we add a specific amount of salt, sodium chloride, 1M NaOCl, to the solution. Now, normally, salt makes bare gold nanoparticles clump together, aggregate. But in this design, when the viral RNA is bound to the aptamer on the AUNP surface, the addition of NaCl actually induces aggregation of the AUNPs only if the RNA is bound in a certain way. This aggregation changes the environment right around the nanoparticles, shifting that surface plasmon resonance we talked about. And finally, that SPR shift becomes visible as that dramatic color change from wine red to bluish gray. It's like a beautifully choreographed molecular dance, leading to a clear visual yes or no.
That really does sound like a masterclass in molecular engineering. Every piece has its specific, elegant job. It's incredible how such complex chemistry can boil down to something so visually simple. But the brilliance here isn't just the chemistry, it's also the engineering, right? Expert, we have this amazing chemistry, but how do you actually package it into a practical device, something useful in the real world, far from a fancy lab? This is where the engineering side really shines, making it accessible.
Indeed. Yeah, that jump from a reaction in a tube to a usable diagnostic tool, that's often the biggest hurdle. And this research tackles that brilliantly with a really smart engineering solution. The device itself has all the characteristics you'd want for something meant for global pandemic response. It's incredibly low cost, it's small, handheld, and crucially, its case is 3D printed. That detail of the 3D printing is huge for accessibility.
Yeah.
It means the physical shelf of the electronics can be made almost anywhere with just a basic 3D printer. That lowers manufacturing barriers, allows for local production. It sort of fosters this open source hardware idea.
Yeah, that 3D printing part alone is a game changer for access and local making. But let's look inside at the electronics. Can you detail the components for us? Why these specific choices? How do they achieve this compact, efficient, affordable system?
Certainly. At its core, the device is elegantly simple but really effective. It's got three main electronic parts. First, the transmitter board, TX. This has narrowband LEDs that shine light at just two very specific wavelengths, 520 nanometers and 630 nanometers. These aren't random. They line up perfectly with those two SBR peaks we talked about, the wine red color peak and the bluish gray color peak. By using only these two wavelengths, they drastically cut down the complexity and cost compared to, say, spectrophotometers that scan a whole range of light. Second, there's the receiver board, RX. This houses the light-to-frequency converter chip, the TCS3200. This little chip is pretty clever. Instead of just measuring how bright the light is, which can be tricky and need calibration, it converts the light intensity into a proportional frequency signal. Think of it like turning light into a digital pulse train. That's much easier for a microcontroller to read accurately without needing complex analog-to-digital converters. This selective measurement at just two wavelengths, using frequency output, dramatically speeds up the reading, simplifies the processing, and cuts down power use. And finally, both these boards talk to a nano microcontroller. That's the brain. It controls the LEDs, reads the frequency from the sensor chip, does the basic math, and then sends the result out via standard USB cable to a computer, or maybe even a simple display later on. Using a nano microcontroller again emphasizes that commitment to low-cost, readily available parts. The whole thing, tucked into its 3D printed case, is designed for maximum efficiency and minimal cost.
Okay, let's really unpack how simple and affordable this is, because the numbers are genuinely, well, astounding. Expert, tell us about the cost for the device itself and for running each test.
The cost effectiveness is honestly one of the most compelling parts of this whole thing. We're talking about the entire device costing less than $20 US to build. That's what, around 1580 Indian rupees for the whole unit. And here's the kicker, the part that truly democratizes testing. The cost per individual test can be less than $1 US, about 80 Indian rupees. Combine that with the speed, less than 5 minutes, for a result from a prepared RNA sample, and you've got something revolutionary. To put that sub $1 cost in perspective, it means making diagnostics genuinely accessible to everyone, everywhere. Think about a remote village where maybe a $50 RT-PCR test is just impossible for most people. But a $1 test, that could be the difference between unchecked spread and timely action. This isn't just cheaper, it's a total paradigm shift for global health equity and how we respond to outbreaks. Decisions can be made instantly, right there at the point of care, no waiting days for lab results, no need for expensive infrastructure.
That's an incredible contrast to what we were dealing with, especially early in the pandemic. Expert, if we connect this back to the bigger picture, how does this device stack up against other rapid detection methods that emerged during the crisis? Lots promise speed, but this seems different in terms of accessibility.
It's a really crucial distinction to make. During the pandemic, we saw this fantastic burst of innovation in rapid diagnostics, which was great. We had lateral flow antigen tests, various electrochemical sensors, graphene sensors, SIRS biosensors, all sorts. And many did offer faster results than RT-PCR, maybe 30 minutes to a couple of hours. But the critical difference, if you connect it to the bigger picture, was often in practical deployment. Many still needed specialized, maybe smaller instruments, or they involved multi-step sample preps that could still be fiddly or take time. Some needed complex nanomaterials that weren't necessarily quick or cheap to make at scale. So they might have been faster than RT-PCR, but they weren't always simple or ultra-affordable for really widespread decentralized use. This handheld device just sidesteps those complexities. It's a single compact unit, minimal training needed, results in under 5 minutes, and at a tiny fraction of the cost. It's a true point-of-care device, designed not just for a clinic, but potentially for a community health worker out in the field, miles from any lab. That simplicity and cost efficiency are where it really stands out. And the measurement technique itself is really ingenious, making it so user-friendly. Like we said, instead of measuring absorbance across a whole spectrum like lab machines, this device just measures light intensity at those two specific wavelengths,
520 millimeters and 630 millimeter, using those narrowband LEDs. That selective measurement is super efficient. Cuts down readout time, simplifies the electronics, uses less power, perfect for a handheld, maybe battery-powered unit. But how do you easily interpret just two frequency readings without needing a scientist? That's where this really clever and user-friendly slope classification method comes in. Imagine you plot 2 points on a graph, one representing the frequency reading at 520 millimeters, the other at 630 millimeters. Then you draw a line between them. If that line slopes upwards, meaning it's a positive slope, the signal at 520 millimeters is lower than at 630 millimeters. That's your clear signal for a COVID-19 positive sample. Remember, the solution turned bluish-gray. That means it absorbed more light at 520 millimeters lower frequency reading and less at 630 millimeters higher frequency reading, hence positive slope. Conversely, if that line slopes downwards, a negative slope, it indicates a COVID-19 negative sample. Or, importantly, a sample positive for something else like dengue, which our specific aptamer won't bind to. No complex math or spectral graphs needed. It's a simple visual yes or no based on the direction of that line. Makes it incredibly easy to interpret for anyone, anywhere.
That slope classification is brilliant. So intuitive, makes it really accessible, and removes the need for complex data crunching. Now let's talk about the validation. How rigorously was this device actually tested? What kind of samples did they use? Under what conditions to prove it works?
Yeah, the researchers definitely put it through its paces to check reliability. They used a set of 15 clinical samples, which is really important for real-world validation. This included 10 samples from COVID-19 patients, 5 confirmed positive, 5 confirmed negative, plus 3 samples from dengue patients and 3 control samples, just the aptamer-coated AUNPs in pure water. And what's really significant is that these samples were tested right after the viral RNA was extracted. No preamplification like PCR, no other pretreatment. That streamlined process is key to the speed. The tests were done under carefully controlled ideal conditions, temperature between 25 and 27 degrees Celsius, and a neutral pH of 7. These conditions are critical for the chemistry to perform reliably, ensuring the nanoparticles and the aptamer-RNA interactions behave exactly as expected.
Okay, and what did those tests show? What were the actual results for the different samples? Did they match up with the science?
The results were remarkably consistent, really clear, and validated the whole mechanism perfectly. For all the COVID-19 positive samples, they consistently saw that visible color change from wine red to bluish gray. And that visual change matched the electronic readings exactly. Low absorbance signal at 520 millimeter, high absorbance signal at 630 millimeters, which, as we discussed, creates that characteristic positive slope output. On the flip side, the COVID-19 negative samples consistently stayed wine red. Electronically, they showed high absorbance at 520 millimeters, low absorbance at 630 millimeters, giving that negative slope. So this clear binary difference confirms the device can reliably distinguish between infected and uninfected samples based purely on whether that target SARS-CoV-2 RNA is there or not.
That's strong confirmation, definitely. But here's where it gets really interesting and touches on something absolutely critical for any diagnostic: selectivity. You need to be sure it's only detecting the thing it's supposed to, right? Not giving false alarms for other common bugs. Expert, how did they check for this? What did they find regarding selectivity?
Selectivity is absolutely paramount. A diagnostic is useless or even dangerous if it's not specific. False positives cause panic, unnecessary isolation, strain resources. False negatives are just as bad. So the researchers very deliberately tested the device against the dengue virus, DENV. Why dengue? Well, it's also a positive sense, a single-stranded RNA virus, quite similar structurally in its genetic material to SARS-CoV-2. That makes it a perfect test case for potential cross-reactivity. If the aptamer wasn't truly specific, a dengue sample might accidentally trigger a positive result. Thankfully, the results were excellent. The dengue virus sample showed no SPR shift whatsoever. They behaved exactly like the COVID-19 negative sample, stayed wine red, produced a negative slope. This strongly confirms the sensor's high selectivity. It's specifically targeting the SARS-CoV-2 and gene RNA, not just any viral RNA floating around. That really minimizes the risk of false positives from other common viruses that might share some superficial similarities. Makes the result much more trustworthy.
High selectivity is a huge plus, ensuring accuracy in a world full of different viruses. Okay, beyond selectivity, there's sensitivity. How well does it pick up different amounts of virus? What's the detection limit? And what does that mean practically?
Another crucial point. The device showed it could reliably tell positive from negative samples down to a level equivalent to a SARS-CoV-2 in-gene CT value of 25 from RT-PCR. Now, for those not familiar, the CT value cycle threshold basically indicates how much virus is there. A lower CT means more virus. A CD25 generally corresponds to a medium viral load, so the device is effective at detecting infections when people are likely most infectious. But this does raise an important question. What about samples with lower viral loads? Higher CT values, maybe above 35, like in very early infection or as someone's recovering. The paper acknowledges this and cleverly proposes a method for extrapolation. By carefully establishing a baseline and correlating standard CD values from RT-PCR with the frequency outputs from their device, they could see a clear trend. He suggests this trend could potentially be used to estimate results for samples with these higher CT values, essentially extending the device's detection range. It's a smart way to potentially push the sensitivity boundary for detecting earlier infections or lower viral levels, making it even more clinically useful. And it's worth stressing again why those controlled conditions, 25 to 27 degrees C, neutral pH 7, are so vital for optimal sensing. These aren't just, fussy lab preferences. They're fundamental to getting consistent, accurate results. Temperature, for example, directly affects the gold nanoparticle state. You need them nicely dispersed until the RNA binds and the salt is added to trigger aggregation. If they clump too early because it's too hot or cold, the whole detection fails. Similarly, the aptamer itself needs the right temperature to bind properly to the gold and then to the RNA target. And viral RNA itself is quite fragile. It can break down quickly at the wrong temperature or pH, making it undetectable. So neutral pH helps keep the RNA stable in the buffer solution long enough for the test to work. These conditions are basically essential guardians of the test's accuracy.
Makes perfect sense. Those controls are key for such a sensitive molecular test. But what's truly fascinating here, and maybe the most powerful aspect for long-term pandemic readiness, is how they thought ahead about evolving viruses. This paper came out around the time Omicron was emerging. What did the researchers do about new variants? How does this design ensure it doesn't become obsolete the minute a new variant pops up?
This is really where the platform's adaptability shines. It shows this profound forward thinking. They didn't just build a test for that version of COVID-19. They built a framework that can adapt. When Omicron emerged, with all its mutations, they immediately did some theoretical work, some bioinformatics, to design a new aptamer specifically for it. They looked up Omicron's spike glycoprotein sequence in GenBank, that huge public database of genetic codes. Then they compared it, aligned it, against the Delta variant and the original Wuhan strain, using tools like NCBI BLAST, it's like molecular detective work, looking for the key differences. And their analysis found something interesting. While there was like 98% similarity overall, they pinpointed this specific gap of 21 nucleotides in the alignment. It was consistently there when comparing Omicron to the others. This 21 nucleotide gap became like a unique genetic fingerprint for Omicron in that region, so that became their new target. They then used a tool to design a new aptamer sequence that was perfectly complementary, a perfect match to that specific Omicron spike sequence. Finally, they used another tool, a LIGO analyzer, to predict things like how well this new aptamer would bind and how stable it would be, even before making it in the lab. The profound implication here is huge. It means this detection device can be adapted for new variants, or even potentially entirely different viruses, just by designing and plugging in a new specific aptamer sequence. You don't need to redesign the whole device, the gold synthesis, the electronics, none of that. It's incredibly modular. This ensures it stays relevant, it stays useful, making it an invaluable tool for ongoing pandemic preparedness. We can pivot to new threats much faster. It means we're not just building tools to fight the last pandemic, we're building tools that can adapt for future ones too.
Wow. Yeah, that adaptability really brings it all together. It paints such a compelling picture of what true pandemic preparedness could look like. Okay, so let's quickly recap the key takeaways from this deep dive into some really groundbreaking research. First, we highlighted that urgent, ongoing need for rapid, low-cost, point-of-care tests, a hard lesson from COVID-19. Second, we explored that novel way of making gold nanoparticles using reusable solid tin, a potential game changer, faster, cheaper, greener. Third, we saw the power of aptamers, selective, stable, rapidly made, offering big advantages over antibodies, especially in tough environments. Fourth, we marveled at the device itself. Handheld, 3D printed, under $20 to build, tests under $1, results in under 5 minutes using that clever color shift and slope reading. And finally, that incredible foresight. The ability to adapt the system for new variants, like Omicron, just by swapping the aptamer, ensures long-term relevance. So what does this all mean for you listening? It means scientific innovation isn't just happening in some far-off lab. It's actively tackling real-world problems, pushing the boundaries of public health. This research gives us a powerful glimpse into how science can fight global health challenges with solutions that are not only effective, but also incredibly accessible and fair. It's like a blueprint for democratizing diagnostics, boosting our collective readiness for whatever comes next, turning uncertainty into informed action.
Exactly. And if we connect this to the bigger picture, this work isn't just about one COVID test. It really is a blueprint for potential shift in rapid diagnostics. The core innovations that reusable tin synthesis for nanoparticles, fast, scalable, eco-friendly, and the adaptable aptamer sensing platform. They show a clear path for detecting all sorts of things. So here's something to think about. Imagine the potential for this low-cost, rapid platform beyond SARS-CoV-2. What other pathogens, flu, malaria, you name it, or even other critical things, maybe environmental pollutants, maybe food safety checks? What else could this be tailored to detect quickly and cheaply in the future? The possibilities are truly immense. It shows how fundamental science breakthroughs can ripple outwards.
A really powerful thought to leave you with. That's all the time we have for this deep dive. Thanks so much for joining us and for your continued curiosity. Until next time, stay curious and keep digging deeper.